Hi, this video is to explain about cloning vector. Cloning vector is a small DNA molecule that carry gene of interest into a host cell in genetic engineering or in cloning. A vector have two or more restriction sites, or we call it as multiple cloning site, MCS. A cloning vector must have origin of replication, or we use the term origin of replication. Why? Because for them to have an origin of replication means they able to replicate by themselves. They don't need the DNA, because we know that bacteria, they have a DNA. This is the DNA in the nucleoid, and then they have another smaller DNA which is called the plasmid. So this DNA will be replicated, and at the same time this plasmid, this DNA also need to be replicated so that their daughter cell will have the same genetic and amount of gene or DNA. So this plasmid can replicate automatically or freely without the assistance of this DNA replication. A vector also need to have selective or selection of marker genes. This is very important for screening purposes. I will explain that later. How a cloning vector help us? Because a vector can be inserted into a host cell very easily, and then they can survive in the cell, and then they can express the gene that we include or inserted into the cell. This is a plasmid, carries certain gene. When we insert the gene of interest and then they undergo protein synthesis, okay, this gene interest will be able to be expressed, meaning that we can produce the protein that encoded by this gene of interest. So the plasmid is will be able to carry and then express the gene. There are 4 types of cloning vector that you should know. First one is the plasmid, the most commonly used, the bacteriophage, COSMID, and then yeast artificial chromosome. The example of a plasmid is PUC18. Plasmid is a small circular ring, circular bacterial DNA. Circular by means that the beginning meets the end, and then it is ring shaped. It is not part of bacterial chromosome, but it also carries certain gene. They can replicate freely. Plasmid can replicate freely because it has origin of replication. And plasmid able to carry foreign DNA. Bacteriophage is a virus that infects the bacteria. It's also known as phage and can carry almost 20 kilobases of base pairs of foreign DNA. COSMID is a bacterial plasmid which is inserted with COS gene. COS gene originated from bacteriophage, lambda phage. It can carry up to 44 more sequence of gene. PHAGE. Example of yeast artificial chromosome is PHAGE. They are unicellular fungi. They are very easy to grow and also have plasmid. And they can carry more than 100 kilobases, up to 1000 kilobases. So basic characteristics of a successful cloning vector. First, they must be able to accept foreign DNA. And this foreign DNA can be inserted into restriction sites. Because they contain multiple cloning sites, so they can be inserted to any multiple cloning site. But even though I am saying any, the multiple cloning site or the must contain restriction site that is similar to the sequence used to cut the gene of interest. Meaning, if of interest is cut using the ECOR1, so the restriction sites of the cloning vector must be cut using the similar enzyme ECOR1 also. So multiple cloning site is actually a short segment of DNA that contain many restriction sites. So this is where many types of restriction enzyme can recognize and then cut. So many foreign DNA fragments can be used. For example, this one, ECOR1 we know GAATTC, small one, BAM H1, semua tu ada, CCC, GGG. So in this area there are a few restriction site. If we use ECOR1, then ECOR1 will cut it here. If we use BAM H1, it will cut here. Small one, it cut it here. There are a few more. There are so many. There are numerous numbers of restriction enzyme yang kita ingat ECOR1, BAMH1, SMAR1, ada HIN1, SV. Ni I don't know how to pronounce it. The second characteristics that cloning vector needs is able to replicate freely because they contain origin of replication. If not, how can they express the gene that they carry? They also need to possess selectable genetic marker. The most commonly used genetic marker, especially for you to memorize, is AMPR gene. AMP means ampicillin, ampicillin, R for resistance. Ampicillin is a type of antibiotic. So when a bacteria has this kind of gene, meaning they will be able to resist or survive the antibiotic. Okay, antibiotic is used to kill bacteria. If they can resist the antibiotic, then they won't be killed. While LAC gene is gene that encode for beta-galactosidase, as you have learned in topic 6. This selectable genetic marker will help us to identify those plasmid or those cells that carry the vector with the gene of interest. Okay, I will explain it later on. So it will be easy to be selected. Next is characteristics of able to